All right, our next speaker to continue this is going to be Yu-Kai Huang. He is a PhD candidate at UC Irvine, here to tell us about his research.
So thank you for the introduction, and hello everyone. I'm Yu-Kai Huang from UC Irvine, and it's a great honor and pleasure to be here and share my work about determining individual mitochondria metabolic state after mitochondrial transfer. So to talk about mitochondrial transfer, it has been discovered ever since 2004 that cells have the ability to exchange organelles between different cells. And so this is a figure from a review paper. If you look into mitochondrial transfer studies, you can actually categorize them into 4 steps. And first of all, how they isolate out the mitochondria from donor cells. And then second of all, how they transfer or transplant them in vivo or in situ. But here comes the third and the fourth step, which is the quantification of mitochondria inside the recipient cells. You can see you can use confocal, you can use flow cytometry, electron microscopy. And then fourth of all, you evaluate the effect of mitochondrial transfer affecting the recipient cells. So there's a whole lot of assays that you can do to these, but then by diverse methods of these quantification actually causes controversial results. So, good. Okay. Spoil. Okay, so some in the breast cancer research, some groups support that oxalose has increased after mitochondrial transfer and hence favors the cancer cell growth. Others actually claim that found there's a decrease in oxalose and inhibits proliferation. So these are actually very opposite findings. We ourselves, we're an imaging lab. We also try to do this by quantifying. We fluorescently labeled mitochondria. So the local mitochondria inside breast cancer cells, we labeled it with COX-8-GFP, and the isolated ones, we labeled it with Mito7 and Ruby. So we co-cultured them together, and let's look at the first upper panel here. After 12 hours, you can still see between the two colors difference. You can still see between endogenous, the green, and the exogenous mitochondria. But if we wait until 24 hours of incubation, there's actually a huge significance of co-localization, and that is seen to be the fusion of exogenous and endogenous mitochondria. So we assume that this process actually lets the final product inherit the functioning mitochondrial complex. So here comes the question: does this actually enhance oxalose metabolism? We need a method that can actually see mitochondria and then quantify the metabolic state. Okay, I'll wait for a bit.
Oh boy, just use the arrows. This is my fault, not his fault. It's my fault.
So for this talk, you must be listening to a lot of NADH. So what we introduce is the phasor approach to NADH FLIM analysis. So here we have a bright field image of this is a breast cancer spheroid, and we have fibroblasts. If we excite it with 2 photon excitation at around 740 nanometers, voila, you can get the NADH fluorescence. This is called fluorescence imaging. But if you want to do lifetime, you need to synchronize the light modulation and then calculate out capturing the photon arrival time. And then that makes this exponential decay. But what the phasor approach does, it actually Fourier transforms this exponential decay into a new phasor coordinate, which we call the phasor approach. And fortunately, to answer Patrick's answer, why we are focusing more on NADH is that it has a relatively long lifetime difference between free and bound NADH. So bound NADH, it happens when actually it depends on what kind of enzyme it attaches to. So normally when you're in a more oxidative phosphorylation metabolism, you can bind to mitochondrial complexes, and that gives around tau of 3.2 nanoseconds. And if you're at the free NADH, it's at 0.4. So this is relatively a long lifetime difference already. And free NADH is mostly produced in the Krebs cycle, which is the glycolysis. So through that, if you put the two extreme points on the phasor plot and you have your image in between the distribution, you can form a metabolic trajectory. And what happens is, if you have a color map on top of that trajectory, you can give out this beautiful figure over here, the bound NADH fraction. So through this, we can actually tell that this breast cancer spheroid here has a more bound fraction of NADH, indicating that it's a more oxidized state than the fibroblasts. Okay, so the phasor approach is actually not that new of a method, because throughout the years we've been using this approach to measure different metabolic states of different cell lines. We have germ cells, glioblastoma, and breast cancer cells, but then it only restricts on the single cell scale. So in my project, I want to focus on single mitochondria. So what I developed here is what I just call it myself, the FLIM intensity-based image segmentation, called PHEBES. So when you're measuring FLIM on mitochondria, here are some problems that you would face. So when you're exciting NADH through frames to frames, the mitochondrial movement actually causes this motion blur at the final sum to FLIM image. And second of all, if you have, because your NADH intensity is really different depending on what kind of NADH you excite. So if there's a high intensity object inside your image, that actually diminishes other fluorescence signal. Also, NADH is a low signal to noise ratio, but then using two photon excitation and you sum those frames together, you can still get a high quantum yield of it. And final of all, NADH is an intrinsic fluorescent probe, meaning that it's label-free, that we all know about it. So here's an example of a raw flame intensity having a locally high intensity object right over there where the arrow points. And so if you use traditional O2 threshold segmentation, you can only get that part. But any mitochondria that were existing here, you cannot get it. So this is the graphic user interface that I introduced. I will not, if I still have time to be given, okay. So the flowchart of PHEBES, actually, when you're doing throughout the frames, I actually highlighted all of the intensity pixels throughout all the frames, but then that will actually unavoidably amplify the artificial frequency noise across the images. So what we first do, we put in the raw intensity, normalize it, and then if we have this frequency peak noise detected on top of it, we will have to do a denoise process. And then final of all, we also do mitochondria size, because you also might have the blurriness of the mitochondria within the object, or other noises happening. So there's an erosion iteration, and then that you can get to the final image. Here's an example of the first tab. We have we can load the images here, and here are your file names, the image, the original image here, and here's the final image of PHEBES done. So not just this part is also divided into zero mitochondrial objects, but other inside the cell is also seen. Second of all, we have the parameter tab. If you're not satisfied with your image, you can always do other regulations on the parameters. Also, there's a similarity test, which I will introduce later. And the third tab, we have this phasor plot. So my algorithm has also this grouping function. So we can actually group your files together and then plot all the same groups with the same color. And that way you can tell what the distribution it has on the phasor plot. And finally, as PhD students, what we want is a really beautiful image and a really robust method of creating graphs, statistical graphs. So we have this pseudo-colored image here, and also created graph box plots inside. And in the near future, so right now we only have the free-to-bound NADH fraction analysis. I will be so-calling FB fraction afterwards. In the near future, currently I'm still developing other morphology, such as mitochondrial numbers, the length, and the branching, which is we call the solidity of the mitochondria. So what the image similarity does, we are actually exciting NADH without labeling the mitochondria with any fluorescence labels. So we need to make sure that these NADH actually come from mitochondria. So what we did, we compared the Phoebe's functions image with fluorescent labeled image with mito7 and rubies. These were taken with host cells that have uptaken the isolated mitochondria. So you can see through this box area. So this is what I call, if you have the motion blur of mitochondria, this is what we will get. You use a traditional segmentation method, you will get a huge object of the motion blur. But after Phoebe's, you can expand this high resolution into that object and then segment out the correct number of mitochondria. So Phoebe's increases the 35% similarity referenced with fluorescent tagged mitochondria image. And next of all, yes, this is the Phoebe's FB fraction. So normally, if you use the phasor plot, each coordinate represents 1 pixel, but for Phoebe's, each coordinate represents 1 mitochondria. So that way you can know where it lands on the phasor plot. And so we tested our results with 2 metabolic inhibitors. One is the 2-deoxyglucose, which inhibits the glycolysis, making the cancer cell move to a more bound NADH, and the oxalase inhibitor rotenone, which lets the cancer cells move to the free. So looking at adding in mitochondria, we can tell that it moves to the bound NADH. So it really indicates that it has more reliance on oxalase. So all of these were taken from raw NADH intensity, which I want to show here. And so not only beautiful pictures, we need statistical analysis too. So I know the font is quite small, but the arrows here are the ones added with mitochondria, and then here are the controls. We have found there's an increase of bound fraction NADH in both breast cancer cell lines, MCF7s and 231s. So these results were also supported with seahorse assay results, which is the gold standard of metabolic assays so far. If we look at the oxygen consumption rate of MCF7s, there's a significant increase. But then looking at 231s, this is the most interesting part that I want to explain also, is that there is actually no significant difference found in 231s. But there is actually a slight difference here in the bound fraction. So reading into other works done on this triple negative breast cancer cell. So 231s are actually a really aggressive phenotype of breast cancer cell. And researchers have been debating on whether all cancer cells follow the Warburg effect, like they're depending on glycolysis as their main ATP production. So what we want to explain is that if you look at the 231's OCR, which is around 30, and it is already relatively higher than MCS7. So if you are a really aggressive phenotype of breast cancer cell, then you might rely more on the oxalate state, which is supported in the bound NADH. The 231s, which is 0.5, is higher than the MCF7s too. So this really needs to be is an interesting question to answer. Also, if we have other approaches to quantify it. So to conclude my work, Phoebe successfully determines the mitochondrial objects through NADH intensity, and it serves as an integrated method of both mitochondrial image and metabolic index mapping. And the FB fraction of NADH shows that there's a metabolic reliance towards oxalase after mitochondrial transfer. So that actually answers the first slide of the controversial results. And finally, we proved our assumption that transfer mitochondria fuses with endogenous mitochondria and hence inherits the normal respiration function. Future works, we want to compare our Phoebe's algorithm morphology dynamics with mitometer, which is also done by a colleague in my lab. But this mitometer requires the labeling of mitochondria, so you still have to stain it or either transfect it. And ultimately, we wish to apply this algorithm to other mitochondrial dysfunction in diseases such as Alzheimer's, diabetes, and cardiac, or even neural diseases. Here are my references, and I would like to thank my PI Dr. Michelle Digman and Dr. Enrico Bertone, they lead the LFD, which is an imaging center back in UC Irvine. And I would like to thank Dr. Gregory Brewer, who kindly provided his CFIRS assay result for me to present here. And the mitometer, this whole tracking concept actually came from Dr. Austin Lefebvre. So he's working now in Calico, which is a Google-funded life science company. So thank him for this whole idea, and then let me have this opportunity to come here and share our works. And I would like to thank all of the members of Laboratory for Fluorescence Dynamics. By that, I would like to answer any questions you have. Thank you.
Any questions?
How long were the integration times for the NADH images, and in terms of the motion blur that you talked about, and if you don't have the macro time, you're aggregating all of the photons for that integration time, whatever it may be, and that affects the phasor signature of that pixel. If most of that time it's like mitochondria is absent from that pixel, you're basically measuring cytosol, but then for a fraction of that integration time, if the mitochondria is present there, then the phasor signature is mostly that of the cytosol, not of the mitochondria. Does that affect your tracking of mitochondria?
Thank you. Interesting question. So the question was the integration time of the frames that I have collected to gain enough photon counts. So to answer that question, I have, so to avoid the mitochondrial movement, right? I have actually limited my frames to 15 frames. That is around 1 minute. And so for the similarity test, there are also some similarity tests that are below 50%, and that is caused by some mitochondria that are moving really fast. So by that, some might still fail, but then most mitochondria just wiggle around inside that certain area. So that's the main advantage of still using this. Yeah, so not a lot of mitochondria move that much inside the image. I remember there's another question at the back.
Yeah, I was just curious about, because you were imaging the MDM231 cells versus the MCF7 cells. Did you find it harder to image the mitochondria on the MDA cells rather than the MCF7 cells? Because I know they're thinner, so they're like harder to focus on, especially when imaging the mitochondria.
Yes. So her question was whether it was more difficult to image 231 cells mitochondria. And I would say yes. The cell thickness of both, 231s are really thin. So we had to, of course, we use 2-photon excitation, which is not very healthy for the cells. So we limited our pixel dwell time also, but it's still kind of fine because their morphology are sort of more elongated compared to MCS7s, and that actually produces a high area of NADH intensity areas already. So still, we can get enough photon counts through that. Yeah, but I would say breast cancer cells are really a pain to image because I have also done macrophages, and they're really nice because no matter how long you image it, it still stays there, and it's really healthy also.
Do you have any other questions? Any questions from the team?
Yeah, I got a question. So do you think if you added more mitochondria to the 231s, do you think you could raise the OCR at the end, or do you think that you kind of reached a physiologic limit and that's the reason you're not seeing any more change from it?
Yes, so I did not actually mention how much mitochondria are inserted inside the breast cancer cells, but that is really a critical question also, because Caicedo et al., his paper in 2015, he has already done the different amount of mitochondria inserted into breast cancer cells, and he has found around 10 micrograms. That's the limit, the saturation of the OCR that it will increase up to. So I followed his protocol and inserted around 5 micrograms, yeah, to make a good significant increase.
Yeah, you said you were developing the software. Is it available anywhere, or are you still like, do you just have it still in like development mode?
Yes, this is ready to share to the world, but my PI actually told me to share it until we publish it. So this is still under review in Nature, so we hope to let it happen soon also.
Thank you.
If there's no other questions, can I do another small advertisement? So if you're really interested in this talk, you can participate in the 16th LFD workshop, which is in UC, University of California, Irvine. We have a lot of different microscopes that do fluorescence dynamics, and it is really interesting. I will be one of the tutorials to give the experiments demos. If you're interested, you can contact me after this talk.
All right, you're supposed to clear your plugs. I will allow it. Thank you.
Thank you.
Thank you for the presentation, and thanks to our presenter again. Thank you.
All right, and our final